you've done some of the experiments to show, for example, that USB30 inhibition can affect mitophagy in the brain. With we worked together on that one, and you had previously independently shown the effects in the heart. And it seems like the effects are maybe a little bit different in the different tissues. Any thoughts on that?
Yeah, so I think this is a really interesting topic because, I mean, in different tissues, there's different levels of mitochondrial mass, right? And also dynamics and mitochondrial mass and also mitochondrial metabolism will be totally different among all the tissues. And maybe in a same tissue in different areas.
Yeah, I mean, looking at the heart, so depending on who you talk to or which paper you look at, it's like a third of the volume of the heart cells, the myocardial cells, are mitochondria, right? So it's
it is. As actually roughly like 30% of the cell volume, cardiomyocyte volume, has been occupied by mitochondria, and we do need mitochondria to produce large amount of ATP to support the contraction and the relaxation. Is another interesting part is the metabolism of the mitochondria is very dynamic, right? Under different conditions in the physiological settings, the mitochondria function and metabolism have been changing all the time. So we believe mitophagy is required to adapt to the new condition and to adapt to the new environment in any kind of tissues, including the brain, the heart, and the muscle. But because the heart, it's a metabolism is very different with other tissues. It utilizes a lot of fatty acid, glucose, and can change during exercise or during disease conditions. That's why I think autophagy has been so critical to the cardiac function, because it's the mass, the volume, their numbers, and also their metabolism.
I'm a neuroscientist by training, so cardiac has not been the top of my list, but just looking at obviously mitochondrial function, it's hard to ignore the heart just because it is such an energetically demanding tissue. And also last year, this paper came out showing that if you take out USB30 or knock down USB30, that you're able to reduce the cellular senescence in the heart, suggesting that if you have more mitophagy, it might reduce senescence that comes with aging. Now, this was an accelerated aging, a DGAL model. So we're working on kind of detangling how relevant that is just to aging. But we know that mitochondria kind of become more dysfunctional with the process of aging. We know that clearing these damaged mitochondria should be beneficial and help with the senescence in the peripheral tissues, and then also neurodegeneration in the brain. Are you guys seeing kind of similar things with enhancement of mitophagy having potential for cardiovascular diseases?
That's actually yes. So we are doing some mitochondria defect in the heart diseases, and we found USB30 inhibition can augment mitophagy in the heart and also can mitigate the cardiac dysfunction in these mitochondria diseases. We also trying to set up experiments using surgical models to induce pressure overload models in the heart, and trying to use USB30 inhibitors and also mitophagy method to rescue this kind of heart failure.
You have some very nice manuscripts that we will link here as well, if anybody else wants to have a look. And thank you so much for joining us. I'm gonna
thank you so much.